Chapter 2, Basic Cell Biology. In this chapter, image. Chapter 2, Basic Cell Biology. In this chapter, image. Getting to know the cell. Image. Understanding the basics of chromosomes, DNA, and genes. Image. Exploring simple cell division. Image. Appreciating the complexities of meiosis. Genetics is closely related to the study of how cells work. The process of passing genetic material from one generation to the next depends completely on how cells grow and divide. Organisms such as bacteria or yeast that reproduce asexually simply copy their DNA through a process called replication, which is covered later in Chapter 7 link, and split in two. But organisms that reproduce asexually go through a complicated dance that includes mixing and matching strands of DNA, a process called recombination, and then having the amount of DNA for special sex cells that join to create completely new genetic combinations for their offspring. These amazing processes are part of what makes you unique from every other human on the planet. In this chapter, we provide a brief introduction to cell structure, DNA, and chromosomes. In addition, you need to be familiar with the processes of mitosis, cell division, and meiosis, the production of sex cells, to appreciate how genetics works. So come inside your cell and let us introduce you to the basics. Later in this book, we will spend more time on the details of DNA and chromosomes, since these topics lay the groundwork for all things in genetics. Entering the world of the cell, there are two basic kinds of cell types, distinguished by whether or not they have a nucleus, a compartment filled with DNA surrounded by a membrane. Prokaryotes, organisms whose cells lack a nucleus and therefore have DNA floating loosely in the liquid center of the cell. Eukaryotes, organisms that have a well-defined nucleus to house and protect the DNA. The basic qualities of prokaryotes and eukaryotes are similar but not identical, because all living things fall into one of these two groups. Understanding the differences and similarities between cell types is important. In this section, you will learn how to distinguish the two kinds of cells from each other, and you will get a quick tour of the insides of cells, both with and without nuclei, plural of nucleus. Figure 2.1 link shows you the structure of each type of cell. Cells without a nucleus. Scientists classify organisms composed of cells without nuclei as prokaryotes, which means before nucleus. The prokaryotes you've probably heard of are bacteria, which are mostly single-celled organisms. Their cells lack nuclei and have relatively small amounts of DNA. You can flip to Chapter 8 link for more on the amounts of DNA different organisms possess. Prokaryotes are the most common forms of life on Earth. They exist in every type of environment, including within and on other living things. In fact, at this very moment, trillions of bacterial cells live in and on your body. Most of the bacteria in your body are completely harmless, and others are beneficial to your health. For example, the digestion going on in your intestines is partially powered by bacteria that break down the food you eat. The bacteria that make news headlines, however, are the small percentage that can be deadly, causing rapidly transmitted diseases such as cholera, a severe intestinal disease found most often in populations without a clean supply of water. The vast majority of prokaryotic cells are beneficial to human life and the ecosystems we live in, performing vital functions such as photosynthesis and recycling the dead into usable nutrients for other forms of life. In fact, it's not too much to say that no other forms of life would survive for long on the planet without the prokaryotes. The schematic illustration of the basic structure of a prokaryotic cell. The part A image on the top and it contains few callouts are plasma membrane, cell wall, ribosomes and DNA. The part B image in the bottom it contains few callouts are cilia, mitochondrion, ribosomes, smooth endoplasmic reticulum, lysosome, centriole, plasma membrane, Golgi apparatus, rough endoplasmic reticulum, vacuole, nucleolus nucleus, vesicle formation and cytoplasm. Image, figure 2.1, link, a prokaryotic cell, A, is very simple compared to a eukaryotic cell, B. Note that these cells are not drawn to scale. Prokaryotic cells are normally 10-100 times smaller than eukaryotic cells. The basic structure of a prokaryotic cell is shown in figure 2.1 link. Scientists call the boundary that separates all cells from their environment the plasma membrane. Membranes are thin, flexible sheets of molecules that act as a barrier. The plasma membrane regulates the exchange of nutrients, water, and gases between cells and their environment. In addition to the plasma membrane, Many prokaryotic cells are surrounded by a cell wall that primarily serves as protection from pressure changes caused by movement of water into and out of the cell. The liquid interior of the cell is called the cytoplasm. The cytoplasm provides a cushiony, watery home for the DNA and other cell machinery that carry out the business of living. Prokaryotes typically have a single hoop-shaped piece of DNA that floats in the cytoplasm. Scientists call the pieces of DNA inside cells chromosomes. See the section, Examining the Basics of Chromosomes link, later in the chapter. Cells with a nucleus. Scientists classify organisms that have cells with nuclei as eukaryotes, which means true nucleus. Eukaryotes range in complexity from simple, single-celled organisms to complex, multicellular organisms like Eukaryotic cells are fairly complicated and have numerous parts to keep track of. Refer to Figure 2.1 link. Like prokaryotes, eukaryotic cells have a selective border called a plasma membrane, and sometimes a cell wall surrounds the membrane. Plants, for example, have cell walls, but that's where the similarities end. Remember image. The distinctive feature of the eukaryotic cell is the nucleus, the membrane-surrounded compartment that houses the DNA. The nucleus protects the DNA from damage during day-to-day living. The DNA of eukaryotes is divided into separate pieces called chromosomes. Eukaryotic chromosomes are usually long, string-like segments of DNA instead of the hoop-shaped ones found in prokaryotes. Another hallmark of eukaryotes is the way the DNA is packaged. Eukaryotes usually have much larger amounts of DNA than prokaryotes, and to fit all that DNA into the tiny cell nucleus, it must be tightly wound around special proteins. For all the details about DNA packaging in eukaryotes, flip to Chapter 6 link. Unlike prokaryotes, eukaryotes have all sorts of cell parts, called organelles, that perform specific functions for the cell. Membranes form the boundaries of the organelles. Two of the most important organelles found in the cytoplasm are mitochondria, the powerhouses of the eukaryotic cell. Mitochondria pump out energy by transferring energy from food to ATP, adenosine triphosphate. ATP acts like a battery of sorts, storing energy until it's needed for day-to-day living. Almost all eukaryotes have mitochondria, chloroplasts, found in plants and algae. Chloroplasts capture energy from the sun and store it in sugars they make from carbon dioxide and water. These sugars can be used as a source of energy for plants, or for organisms that eat the plants. In most multicellular eukaryotes, cells come in two basic varieties: body cells, called somatic cells, or sex cells, called gametes. The two cell types have different functions and are produced in different ways. Somatic cells. Somatic cells are produced by a type of cell division called mitosis. See the section, Mitosis, Splitting Up link, for details. Somatic cells of multicellular organisms like humans are differentiated into special cell types. Skin cells and muscle cells are both somatic cells, for instance. But if you were to examine your skin cells under a microscope and compare them with your muscle cells, you'd see that their structures are very different. The various cells that make up your body all have the same basic components: membrane, organelles, and so on, but the arrangements of the elements change from one cell type to the next so that they can carry out various jobs such as digestion, intestinal cells, energy storage, fat cells, or oxygen transport to your tissues, blood cells. Changes that occur only in the DNA of somatic cells, that is, changes that occur over the course of a person's lifetime, cannot be passed from parent to child. Gametes. Gametes are specialized cells produced by a type of cell division called meiosis for sexual reproduction, as shown in the human life cycle in Figure 2.2 link. Sexual reproduction combines genetic material from two organisms and requires special preparation in the form of a reduction in the amount of genetic material allocated to gametes. See meiosis, making cells for sexual reproduction link, later in the chapter for an explanation. Only eukaryotic organisms engage in sexual reproduction, which we cover in detail at the end of this chapter in the section, Meiosis in the Human Body link. In humans, the two types of gametes are eggs and sperm. Unlike those in somatic cells, changes in the DNA of gametes can be passed from parent to child. The schematic illustration of the human life cycle depicts a male and female diagram on the bottom left. The two arrows pointed in clockwise direction and have few callouts are meiosis, egg cell, sperm cell, haploid gametes, n equals 23, fertilization, diploid zygote, 2n equals 46, mitosis and development n multicellular diploid adults, 2n equals 46, image. Figure 2.2. Link. The human life cycle. Exploring your DNA. Chromosomes and genes. The nucleus of a eukaryotic organism is home to most of its DNA. The mitochondria and chloroplasts also have DNA, which we discuss more in Chapter 5. Link. In humans, nearly all the body's cells have a nucleus filled with DNA. Mature red blood cells do not. Each of your cells contains 2 copies of your genome. That is, all the DNA you inherited from your mother, minus the mitochondrial DNA and all the DNA you inherited from your father. Your genome contains all the information necessary for the human body to grow and develop and for the various parts of the body to function every day. The amount of information carried in your DNA is staggering. It is basically the how-to guide for your body. DNA is generally measured in base pairs, which we discuss in detail in Chapter 5. Link. Your genome has approximately 3 billion base pairs. The 2 copies of your genome add up to 6 billion base pairs per cell, which means the DNA from a single cell would measure just over 6 feet, or 1.8 meters, if stretched out and laid end to end. In order to organize all that information and to fit it into each of your cells, your DNA is packaged into chromosomes. If you think of your genome as a multi-volume how-to guide, then each chromosome is a single book that belongs to the set. Your genome is contained in 23 chromosomes, and each cell contains 2 sets of those 23 chromosomes, for a total of 46 chromosomes in every cell. Examining the basics of chromosomes. Chromosomes are thread-like strands composed of DNA, to pass genetic traits from one generation to the next. The chromosomes must be copied. See Chapter 7. Link. And then the copies must be divided up into the cells that will form the next generation. Most prokaryotes have only one circular chromosome that, when copied, is passed on to the daughter cells, new cells created by cell division. Eukaryotes have more complex problems to solve, like sorting the chromosomes properly to make gametes for sexual reproduction, and their chromosomes behave differently during mitosis and meiosis. This section gets into the intricacies of chromosomes in eukaryotic cells because they're so complex. Counting out chromosomes. Each eukaryotic organism has a specific number of chromosomes per cell, but it is different for each organism. For example, the human genome is contained in 23 chromosomes, while the fruit fly genome is contained in only 4 chromosomes. Remember that you have 2 copies of every chromosome for a total of 46 per cell. Adult fruit flies would have a total of 8. Chromosomes come in 2 varieties. Sex chromosomes. These chromosomes determine sex, male or female. Human cells contain 2 sex chromosomes. Females should have 2 X chromosomes, while males should have an X chromosome and a Y chromosome. To find out more about how sex is determined by the X and Y chromosomes, flip to Chapter 6. Link. Autosomal chromosomes. Autosomal simply refers to non-sex chromosomes. Sticking with a human example, if you do the math, you can see that humans have 44 autosomal chromosomes. 46 total chromosomes minus 2 sex chromosomes equals 44 autosomal chromosomes. Human chromosomes can be divided into pairs and numbered to create a karyotype like the one pictured in Figure 2.3. Link. You have 22 pairs of uniquely shaped autosomal chromosomes plus 1 pair of sex chromosomes for a total of 23 chromosome pairs. Your autosomal chromosomes are identified by numbers 1 through 22, and are generally arranged based on size, with chromosome 1 being the largest and chromosome 22 being the smallest. So you have 2 chromosome 1s, 2 2s, and so on. Scientists and doctors can learn about certain health conditions by looking at a person's karyotype. You can discover more about karyotyping in Chapter 12. Link. The individual chromosomes in each pair of your chromosomes are homologous, meaning that the paired chromosomes are identical to one another with respect to which genes they carry. In addition, your homologous chromosomes are identical in shape and size. These pairs of chromosomes are sometimes referred to as homologs for short. Chromosome numbers can be a bit confusing. Humans are diploid, meaning we have 2 copies of each chromosome. Some organisms, like bees and wasps, have only 1 set of chromosomes. Cells with 1 set of chromosomes are called haploid. Other organisms have 3, 4, or as many as 16 copies of each chromosome. The number of chromosome sets held by a particular organism is called the ploidy. For more on chromosome numbers, see Chapter 12. Link. The total number of chromosomes doesn't tell you what the ploidy of an organism is. For that reason, the number of chromosomes an organism has is often listed as some multiple of n. A single set of chromosomes referred to by the n is the haploid number. Humans. R2n equals 46. indicating that humans are diploid and their total number of chromosomes is 46. Human sex cells such as eggs or sperm are haploid. Refer to Figure 2.2 link. The schematic illustration of human chromosomes can be divided into pairs and numbered to create a normal karyotype. The image contains 22 pairs of uniquely shaped autosomal chromosomes plus 1 pair of sex chromosomes for a total of 23 chromosome pairs. The autosomal chromosomes are identified by numbers 1 through 22 and are generally arranged based on size, with chromosome 1 being the largest and chromosome 22 being the smallest. Image. Figure 2.3. Link. The 46 human chromosomes are divided into 23 pairs. Technical stuff. Image. Geneticists believe that the homologous pairs of chromosomes in humans started as one set, that is, haploid, and the entire set was duplicated at some point in some distant ancestor, many millions of years ago. Examining chromosome anatomy. Chromosomes are often depicted in stick-like forms, like those you see in Figure 2.4 link. Chromosomes don't look like sticks, though. In fact, most of the time they're loose and string-like. Chromosomes only take on this distinctive shape and form when cell division is about to take place, during metaphase of meiosis or mitosis. They're often drawn this way so that the special characteristics of eukaryotic chromosomes are easier to see. Figure 2.4 link points out the important features of eukaryotic chromosomes. The centromere is the part of the chromosome that appears pinched, while the telomeres are the ends of the chromosomes. Telomeres are made of densely packed DNA and serve to protect the chromosome from being broken down. The schematic illustration of basic structure of eukaryotic chromosomes in stick shape. The call-out point to the important features of eukaryotic chromosomes are telomere, centromere, locus and two sister chromatids. Image. Figure 2.4. Link. Basic structure of eukaryotic chromosomes. Finding your genes. The differences in shapes and sizes of chromosomes are easy to see, but the most important differences between chromosomes are hidden deep inside the DNA. Chromosomes carry genes, sections of DNA that provide the building plans for specific molecules, such as proteins, and that are associated with various traits. The genes tell the body how, when, and where to make all the structures that are necessary for the processes of living. In continuing the how-to guide analogy, each gene could be a chapter with a very specific set of instructions. Your genome contains approximately 22,000 genes, with each chromosome containing a varying number of genes, from fewer than 300 genes on the smallest chromosome, the Y chromosome, to more than 3,000 genes on the largest, chromosome 1. The smallest human genes are only a few hundred base pairs in length. The largest is the gene that encodes the muscle protein dystrophin, which has 2.4 million base pairs. You have 2 copies of each gene, with the exception of genes on the sex chromosomes in males, who have only 1 copy of most genes on the X and Y chromosomes. You inherit 1 copy of each gene from each parent. Each pair of homologous chromosomes carries the same, but not necessarily identical, genes. For example, both chromosomes of a particular homologous pair may contain a gene that controls hair color, but one can be a brown hair version of the gene and the other can be a blonde hair version of the gene. Alternative versions of genes are called alleles. Refer to Figure 2.4 link. One of these alleles would be from your mother and the other from your father. Any given gene can have 1 or more alleles. In Figure 2.4 link, 1 chromosome carries the allele A while its homologue carries the allele A. The relative size of a gene is very small relative to the whole chromosome. The alleles for a given gene are large here so you can see them. The alleles are associated with different physical traits, phenotypes, you see in organisms, like hair color or flower shape. You can find out more about how alleles affect phenotype in Chapter 3 link. Each point along the chromosome is called a locus, Latin for place. The plural of locus is loci, pronounced loci. Most of the phenotypes that you see are produced by multiple genes, that is, genes occurring at different loci and often on different chromosomes acting together. For instance, human eye color is determined by at least 3 genes that reside on 2 different chromosomes. You can find out more about how genes are arranged along chromosomes in Chapter 11 link. Mitosis, splitting up. Most